Okay, everyone. So today we're going to talk about the lac Operon. But before we even start talking about the lac Operon, let's understand something about prokaryotic cells. Well, first thing I'll tell you, why the lac Operon? What's up with this subject? Well, I'll tell you what happened. Before, this subject used to pop up now and then, but it never really came out like on NBME. I never seen it at that level. Like you'll catch it on UWorld. And yes, everybody took the exam, had to learn it pretty well, just in case it ever came out. But don't ever remember anyone telling me, hey, I had lac Operon in my exam. But just recently, the most recent NBME had a question on this. And I was tutoring a student, and he told me he just didn't understand it. So I was explaining it to him, and it was kind of hard for him to understand because they kind of showed the lac Operon from the perspective of a graph. And they asked the question. So we'll post that question up. Like, spoiler alert. We'll just post the question up. We'll take a look at it. We'll analyze it. We should be able to understand it properly. It was a really good question, actually. It was a really good question. It wasn't one of those got you questions. So definitely you'll come off learning about the lac Operon, and we'd have a little system to remember it just in case you forget it, right? Little mnemonic that will tie into it. All right, so let's start off. We're recording already. Let's start. So, okay, first thing we need to understand, bacteria loves to consume glucose. It likes to consume energy in general, right? Not only bacteria, fungus, any kind of microorganism. So bacteria usually has a very specific type of cycle. So let's take a look here. Here you're seeing the life cycle of a bacteria. Lag phase, log phase, stationary, and death. So lag phase, they're just getting ready to basically proliferate. They're getting ready to undergo high levels of proliferation. In log phase, they are proliferating. Stationary phase, they have as much death as they have reproduction. So it kind of plateaus. But then afterwards, they have death. And this is normal in every single microorganism. And, well, this is the problem with this. Some of this log phase can be intensified. So you can intensify the log phase by manipulating the actual environment. So what do I mean? All right, you guys probably know this. If I give a wide spectrum antibiotic, so let's say a patient, she can be prone to have infections like Candida, for example, or she can get C. diff. Many different things can happen. Why? Because you're eliminating the competition. So there's a competition. This is a microbiome that gets formed. These kind of microorganisms live in synchrony. They live together. And everybody is sharing the environment. There's some food available for everyone. So they get to live, but notice they don't over-replicate. They can't over-replicate. They have to maintain their replication at a certain level. When they over-replicate, they cause infections. So there's certain things that I can do in order for them to over-replicate. One thing can be, for example, eliminating the competition, like we just mentioned, giving wide-spectrum antibiotics. Then other microorganisms can now thrive because now they have more resources, more food, more oxygen, more space. That makes them grow. They grow to a certain level where they start to express virulence factors. They start to express exotoxins. They start to express themselves, basically, and they engage with our immune system. And that leads to an infection. Okay? So we also see another mode of them growing, basically. Another mode is giving certain types of nutrients that will enhance their proliferation. Now, certain types of environments favor the growth of certain types of microorganisms. Like what? I'll give you an example. Alkaline environment stimulates Vibrio proliferation. Vibrio likes to grow in alkaline areas, right? Iron. If you are around iron, Yersinia likes to grow around iron environments. So does Listeria, right? Many different types of organisms are favored to grow when you actually promote their favored environment. All right, but in this sense, what we're talking about today is a normal type of growth, a growth that does not override the normal level. This is just a typical growth based on the consumption of glucose and galactose, being very specific, from E. coli. So E. coli, of course, is the star of our show, like always. It's going to be E. coli. So let's take a look. Let's understand what happens here. E. coli. So just a reminder, remember that E. coli is a lactose fermentator, right? It likes to fermentate lactose. McConkey positive. What else can I tell you? Also, it can be found with different subtypes. You have H-pit, hemorrhagic, the pathogenic type, the pediatric type, they also call it, the invasive type, the traveler's diarrhea, the toxogenic. These are like the 4 different main types of E. coli. Now, I wanted to tell you one thing. When we look at fermentators of lactose, you have fast fermentators. And the mnemonic was key, because you need a key in order to be fast, to get into your car and move fast. Who remembers? What are the microorganisms that fermentate lactose rapidly? Does anybody remember? Anybody remember? Type it in if you remember. Klebsiella. Good. What else? Klebsiella, E. coli. I'll give you that one since we're talking about it. And Enterobacter. Good. Yeah. Guys are on fire. You guys know this. Enterobacter. Good. These are the key. So you need a key in order to turn on your car so you can go fast. So these are the fast fermentators. Okay. Just a side note, I can do an indole test. Indole test to see which one of these are going to be E. coli. So I can do this and it should give me positive for E. coli. E. coli will be positive indole. Now, if I want to get a differentiation between H-pit, all of them do fermentation of sucrose, which is sorbitol, except for one of them, except for one. Who remembers? There's one of them that does not. Let me see if you guys know this. I'll be impressed if you guys do. There's one of them that does not. Well, no, out of these are strands of E. coli. You have the hemorrhagic type, you have the pathogenic type, you have the invasive type, and then you have the toxogenic type. So this one is going to be the hemorrhagic type, the 015, the one that causes HUS. That one in specific is the only one that does not fermentate that sorbitol. All the others do. Yes, let me tell you the actual name, the name of this E. coli. This is, yeah, these strains have, I remember I separate them by their abilities to cause problems, like the traveler's diarrhea, 0157. Yes, thank you, Miriam. Definitely, that's that number code, 0157. So yes, the H is basically the H7 strain, 0157H strain. This one is basically the one that has that viral toxin that causes HUS in children. Remember that? But okay, I'm just telling you a quick trick to try to differentiate them just in case. This is sorbitol. They can fermentate that sorbitol. But let's go back to what we were saying. Just a side note here. So going back to what we were saying, we were basically mentioning the fact that in order for them to consume monosaccharides, in order for them to consume their glucose, they need to run a specific type of system. They need to run a specific type of system. It must coordinate with this microorganism. So it is a very interesting feedback system. So first, let's learn the player of the games. What is this? What is all this code? Don't get scared. Don't get confused. It's not too crazy, okay? First thing I would tell you is this. the idea that in the presence of ATP, so let's look at it like this. When ATP is high, this is equivalent to a fed state. This is equivalent to a fed state. This means that the patient has now been introduced to higher levels of glucose, right? When you are high on ATP, this is what happens. When you are very low, you're going to see something that happens within these cells. When the gas tank is on empty, let's say, you will see an increase of cyclic AMP. So let's be clear. Cyclic AMP will increase when there is no presence of glucose, of ATP. And when you are in a full state, you will see a higher level of ATP. It will be increased. All right? That's an important notion to know for now. We'll tell you why exactly down the line. Just remember, this is like the gas tank being on empty. Empty, full. All right? That makes sense so far? So good. Just keep that in mind. Now, we have to understand something. So I'm going to explain this to you. Hopefully it does not get confusing. If it does, let me know, type it in, and I'll explain it again. So we have different players here for this game. This is a regulatory process. This is going to allow E. coli to be able to consume glucose as its main priority, just like us. We prioritize glucose as our major, let's say, monosaccharide that we love to break down in glycolysis and then put into the Krebs cycle in order to extract molecules of NADH, then use the electron transport chain. One thing that you'll see is that E. coli loves glucose. That's this first priority. So in a state, let's say, where glucose is present, let's say you have a high level of glucose. Yeah, let me take a small image and show you guys. Yeah, I have some images here that will look nice. In the presence of glucose, you'll notice that E. coli will be able to break it down very easily, but glucose itself will inhibit adenine cyclase. When you have glucose, you break this down into ATP, and ATP will inhibit adenine cyclase on these type of cells, leading to basically a decrease. You'll have a, hold on, let me put this right. So what you'll have in this sense, if you have a lot of ATP, you will basically inhibit the breakdown. So if you don't have the breakdown, you'll notice that your adenine cyclase will be high because you won't be able to break it down. So yes, you inhibit your adenine cyclase. Adenine cyclase is an enzyme that's responsible for breaking down your ATP. Remember that? You break down your ATP into cyclic AMP. So when you have a lot of ATP, you're able to shut this down. So by shutting it down, you'll have an increase in cyclic AMP. So yeah, do not get confused. I'm going to tell you what happens here. Why is this so confusing? Because a lot of times you'll see they're inhibiting inhibitors and activating an activator. And you'll see a lot of that here. So get your mind straight in that sense. And I'll show you where you'll see that. You'll see this with the repressor when we talk about that. So, okay, first thing I'm just letting you know is that there's a regulatory system that has to do with the presence of glucose. Glucose will give you a higher level of ATP. When you have a lot of ATP, you are at a full state. When you are at a full state, there's a complex here called CAP complex. So I'm going to introduce the players of this game, the regulatory system. Let's name them one by one. So what is this? I'm showing you a piece of the genome of E. coli. Yes, E. coli has genome, genetic material. Unlike us, it's not helical. It's going to be a circular type of DNA. It's a circular type of DNA. And you'll notice that the genomic material here on this DNA, is going to be expressed. So you're going to have expression of this genomic material whenever you are engaging this lac-Operon system. So let's understand. This is just like a magnifying lens to this specific site on the genome. So in this genome site, you have something called LAC-I. LAC-I. So this here has its own enhancer, its own promoter. Everything that it does here is independent to the rest of the system. LAC-I works on its own. Okay? It has one job. LAC-I makes repressors. It makes a specific type of repressor. This repressor will bind this O area, this Operon area, and it turns it off. It turns off the activation of these genes. The activation of these genes. So if we were to talk about the pieces of Operon, the best way to identify them at first approach would be to name these structures. But first, let's divide the structures. I'm going to tell you the structures could be divided by structural type of genes, like you see here, LAC-Z, LAC-Y, LAC-A, LAC-I. I say they are structural because they code, their genetic material will code for specific type of proteins. Okay? So LAC-I, LAC-Z, LAC-Y, and LAC-A. Now, whoever made these initials up was being very generous to us, being very nice. Why do I say that? Because their initials kind of help us out. Well, let's take a look. LAC-I. LAC-I is for inhibitory. Inhibitory, because it makes repressors. It's making a repressor. Remember that the repressor is what will bind that O site, that Operon area. It will bind it to block it. So that's LAC-I. Now, look at LAC-Z. LAC-Z is a gene that will code for a specific type of protein. Specific type of protein called beta galactosidase. So let me put this beta galactosidase here and just put it on the screen like this. Make it a little bigger. So let's explain what in the world is beta galactosidase. Let's understand this. Beta galactosidase will be responsible for two things. Two things. In the presence of lactose, it will break down lactose, which is a disaccharide. Lactose is a disaccharide. It will break it down to galactose and glucose. And it does this through a process of hydrolysis. Hydrolysis. Okay? It'll hydrolyze this reaction. Now, this isn't too different than our system. If you try to remember, think about it, our system, we also make contact with lactose. But instead, we have a brush border enzyme. Instead, we have these brush border enzymes. Brush border enzyme has lactase. Lactase, which can break down disaccharides into the both same type of stuff, same type of deal, glucose and galactose. Remember that some patients, as they get older, they lose the ability to make their lactose. They turn their lactase, they end up turning lactose intolerant, right? That makes sense. But here we're talking about good old E. coli. E. coli doesn't have a problem here. E. coli relies on his Operon system in order to digest lactose. But here's the thing though. It doesn't really care much for the lactose. Its major priority, remember, and I'm going to keep on reminding you, is going to be glucose. Glucose is what it loves to break down. Okay? So here we go. Yes, LAC-Z will basically give you galactosidase. Now you also have LAC-Y. So let's look at LAC-Y. Now, Y is very easy. This one here is like a permease. LAC-Y plays an important type of role, allowing lactose to enter the cell of E. coli. It kind of reminds me of an insulin if you think about it, because it allows this disaccharide to make its way to permeate through the membrane, right? So that's very important there in that step. So it facilitates the transport of lactose to the E. coli. So how do I remember this? Well, look at the Y. If I were to take E. coli, let's say I grab E. coli like this, and I put them really big on the screen, and I draw a Y like this, put a Y. Now I do this, with a little arrow on it. Let's say this right here is lactose, and it's making its way inside. So this Y is like a permease. It allows lactose to join and enter inside E. coli. That little Y shape just gives it up to me in my mind. Reminds me permease activity. All right, next is going to be, so let me put this down here as well, is a lactose permease. Allows it to enter. Then next is LAC-A, the acetylation. Now, this one here is not too important. What it does is that it actually detoxifies. So it's like a cleaning system. That way there's no reducing effect. There's no damage to the E. coli. So this is all just detoxified, basically. So that's the players of this game right now. And I'll also mention something else. When you're able to code for these genes, when you're able to code for them, something that's very important is that you're using mRNA. You're going to use an mRNA type of protein, right? You're going to make an mRNA that will code for all three of them. This is polycistronic activity. So notice the difference when it comes to eukaryotic cells. We are completely different. We make one mRNA for one protein. So for us, one mRNA will be able to make its way out of the nucleus. Remember, you have the capping, poly A tail, splicing, spliceosomes. You get your mRNA out of the nucleus. And when it comes to the ribosomes, you're able to make one protein for one mRNA. Now, prokaryotic cells, first off, they don't have that capping, the poly A tail, or the splicing. They don't have to deal with that at all. And when they make their way out to the cytoplasm, what you'll see is that they can make different proteins with the same mRNA. And that's what you see here. This is an example of that. That's called polycistronic. Polycistronic activity of the prokaryotic cells. So this one mRNA is going to code for all those proteins that we mentioned. For permease, is going to code for beta galactosidase, all of these different types of enzymes, just by having one mRNA. So, okay. We're understanding the players of the game so far, right? Now, here you're seeing the promoter site, the P, the promoter site. This is where your RNA polymerase will bind to, okay? Your RNA polymerase will bind here. Remember that this is a process of transcription. This is not DNA replication. DNA replication needs primers. You need to set down primers to where your RNA is going to bind. You guys remember that. Your DNA polymerases, where they're going to bind. You need to set primers. But here, unlike in DNA replication, the, let's say, RNA will bind to the promoter's sites after the promoters get enhanced by transcription factors. Usually transcription factors will bind the promoter sites, and then RNA polymerase is able to bind to them. Okay? If that's confusing, I'll repeat it. This is the site, this promoter region is going to be able to create a reaction here where you will express transcription factors, and then you'll be able to have RNA polymerase binding to this area. Polymerase. Now, why is it binding here? It's binding here because it wants to transcribe the information that is found on LAC-Z, LAC-Y, LAC-A in order to code for these specific types of proteins that we've just mentioned. Okay, but it can't just bind out of nowhere. It needs enhancement. It needs enhancement. And that's where CAP comes into play. That's where the idea of CAP comes into play. So CAP, for example, this here needs to form a complex. It needs to be able to form a specific type of complex in order for it to activate transcription of mRNA polymerase, in order for mRNA polymerase to code Z, Y, and A. So I'm going to put these together just so you guys won't get confused. This RNA polymerase is specific for Z, Y, A. And in order for this to be able to bind, it needs to have two things. Two things. First of all, you need to take away this repressor. This blockage needs to be removed immediately. It cannot move forward and transcribe if it has a blockage to it. So that makes sense. You have to remove the repressor. That's number one, right? Removal of repressor. All right. Another thing, you need to be basically depleted, because this RNA polymerase will code for tools that you would use to break down your lactose, to make galactose and to make glucose. That's not the primary source of energy. Remember that. We only tap into this source of energy when we don't have glucose in the presence. When we're not in the presence of glucose, now we can consume an alternative type of source, which is galactose, basically derived from lactose. So far, so good, right? Does everybody understand me? If you don't understand, if you have any kind of doubts, you can write them in the chat. I'll explain. So far, so good? Okay, the last part. Okay, so the last part, all I'm telling you is that RNA polymerase is basically, yes. Okay, the repressor, you understand? All I'm going to tell you is this. In order for RNA polymerase to activate itself, first you need to remove the repressor, yes. The second thing that you will need is that you will have to form a specific type of complex. So when you have, let's say, an increase in cyclic AMP, so let's be clear. When you are in starvation mode, like we said here, let me show you this. When you're empty, when you're in starvation mode, let me put here starvation. I'm going to put here full. Yeah, we have it here. It's fed. When you are in starvation, you increase your cyclic AMP. All right? So when you have an increased level of cyclic AMP, that means your glucose is depleted. So let me write it out just so it can make sense. Increase cyclic AMP, that would mean glucose is depleted. If glucose is depleted, now we qualify to consume an alternative source of energy. That alternative source is lactose. So how do we know that it's depleted? Well, for starters, cyclic AMP will increase. Not only will it increase, but it would form a complex. It forms a complex here with CAP. So cyclic AMP and CAP get together. They get together. I'm going to put this here, and they form a complex. Cyclic AMP increase with CAMP. That's the positive stimulator. Does that make sense, Dr. Carey? So we have two different stimulators for RNA polymerase, two different stimulators. One of them is negative. The other one is positive. The positive stimulator is what we just mentioned, a complex of increased cyclic AMP, increase of cyclic AMP with something called CAP. They have to join together. They join together. Now you have proactive stimulation of RNA polymerase in order to make the specific mRNA that codes for LAC-Z, Y, and A proteins. Okay, now negative. You have to remove the repressor. You have to get that repressor out of the way. So I will tell you now how we naturally remove this repressor. How do we get this repressor out of the way? Okay, here goes. So in the presence, and I don't want to confuse anybody, I really don't. So let's pay attention here. It gets a little bit confusing. It does. But I'll tell you what happens here. In the presence of lactose, lactose can become allolactose. Allolactose. Okay? Now, I said it was going to get a bit confusing. This is the part that's a bit confusing. This lactose to allolactose is actually activated by the enzyme beta galactosidase. I know it sounds crazy because that's what we're trying to code. That's what we're trying to make. We're trying to make beta galactosidase. So how in the world is something that we're trying to make, because we don't have it, how is that able to react here and create allolactose? Let me explain. This is a leaky type of activation. I know it's weird. They call this like a leaky stimulation. The presence of beta galactosidase is extremely low. It's not completely gone, but it is extremely low. So E. coli maintains a very low level of beta galactosidase. And this low level of beta galactosidase favors the reaction of conversion of lactose into allolactose. It favors this reaction. This kind of reminds me of enzymes. Try to remember enzymes that deal with catalyzing reactions towards different types of substrates. Like what, for example? I'll tell you. Remember the idea of low Km, high Km? Remember the idea of a patient that is in the presence of a few substrates, therefore the enzyme doesn't activate. That's enzymes with a very high Km. Very high Km will only catalyze a reaction when they're in abundance, they're in the presence of an abundance of substrates. Did that make sense to anybody? Let me know if I lost you there. Something with a high Km usually catalyzes a reaction when it's in an abundance of the substrate. If you just give it a bit of that substrate, it will not react. Kind of like glucokinase. For this reaction, what I'm trying to say, Muskan, for this reaction, you're going to notice that when beta galactosidase is here, acting like it's basically activated through a leaky expression, where it's transcribed at a very, very low level. And at this low level, the reaction that it favors is the reaction that converts lactose into allolactose. Now, let's be clear. Okay, perfect. Good. Once you actually start to transcribe galactosidase at a higher level, you'll notice that it takes a back seat to this reaction and it starts to primarily focus on the hydrolysis of lactose, breaking down lactose for consumption, breaking this down as glucose and galactosidase. So usually the concept of Km and Vmax, usually it's based on different enzymes. But here I'm telling you, this is the same enzyme, but catalyzing different reactions just based on its environment. And at this level, at a low level, it's going to favor a reaction where lactose is turned into allolactose. And it should make sense to us physiologically because this allolactose is what binds the repressor. This is what will bind the repressor. It will remove the repressor. Because once you remove the repressor, now you have removed the negative stimulus. Now you can finally transcribe Z, Y, and A. Does that make sense? Good. So I was using KM as an example. I was just using that as an example so you can know that the manifestation of this enzyme is a bit different when it's at lower levels. But then, yeah, our goal is basically to regulate the system. So our goal is to make E. coli happy. And what makes E. coli happy? First, E. coli wants to eat glucose. He gets to pick what he wants. Yeah, he wants to eat glucose. And that's what he favors. He favors glucose, number one. So in the presence of glucose, he's maximizing replication. He's using the glucose as best he can. He's loving it. Now, once glucose gets very low, let's say there's lactose. He's in the presence of lactose, let's say. In order for him to activate lactose, he's going to need two things. Number one, he needs glucose to be low. These are the prerequisites for the activation. Yes, definitely. These are the prerequisites for the activation of the lac-Aperon system. First of all, he needs to be in what we call, let's say, a starvation phase. I can just say he needs to be empty. He needs to have an increase in cyclic AMP. You see an increase in cyclic AMP when your ATP is low. When your ATP is low, you will see an increase in cyclic AMP. So first off, in order to activate lac-Aperon, first you need increase in cyclic AMP. Why? Because this will form a cyclic AMP and CAP complex. This complex enhances those promoters. And then the enhancement of that promoter will allow RNA polymerase to recognize it, to bind to it, and then to be able to transcribe the genes Z, Y, and A. Right? So this is the positive stimulus. And this here, so just to put it in layman terms, just to put it in normal terminology that we could all understand, increase in cyclic AMP is just, let's say, depletion of ATP. Depletion of ATP. Nice and easy. So you have to be under that. The second thing that he needs, the second thing that will be needed is lactose itself. If lactose is not there, then you cannot activate these genes. So lactose will have to be there because in order for it to be there, it needs to be there in order to turn into allolactose, because allolactose is what binds the repressor. Okay? Negative inhibition. You need to inhibit the inhibitor. You need to inhibit the inhibitor. You need to remove the repressors. And we mentioned what removes the repressors? Allolactose. Good. We're understanding all this. This is awesome. We're now understanding lac-Aperon. There's not much to it. It's not too crazy. Seems pretty simple, right? You guys think you'll do good if I show you the question on the NBME? Let's see. Let's see. Show you guys a question, and then we can go through it. But I want everybody to tell me if you guys understand. If you want me to go through it one more time, I'm willing to do it from the top. So low lactose lead to increase in cyclic AMP. Well, I'll tell you what happens. Good question. Good question. So low glucose and allolactose promote lac-Aperon. Yes, you are right. That's what promotes it. So yes, I don't know how to say your name. I'm just going to say I. So, okay, I'll tell you. That is a good question, and I'll tell you why. Because you'll notice one thing. In order for E. coli to consume lactose, it must break down. It must be able to cause a hydrolysis to the lactose in order to have glucose, then galactose. So you're saying, so low lactose leads to an increase in cyclic AMP? Not necessarily. Low glucose leads to an increase in cyclic AMP. I'll tell you what happens with lactose. You need to be able to cause a process of hydrolysis where you'll break this down into two different monosaccharides, into glucose and galactose. Also remember, this is the same story for all the other types of disaccharides. It's the same idea, for example, with sucrose. Sucrose needs to be broken down. Sucrose needs to be broken down into fructose and glucose. Mannose into glucose and glucose, especially if you want to absorb it in your GI. But what I'm telling you is this. I don't know if I answered your question. So low lactose leads to increase in cyclic AMP. Well, low energy source will lead to an increase in cyclic AMP. Yes, but you can still have a low level of no, you're saying low lactose leads to an increased level of cyclic AMP. That wouldn't make sense. That wouldn't make sense because I'll tell you what, whenever you're in the presence of ATP, you will have a low cyclic AMP, regardless of where it comes from. Does that make sense? Low lactose leads to low conversion of glucose, leads to increase in cyclic AMP. So hold on. Let's get this thought. You're saying low lactose leads to low conversion of glucose. No, I'll tell you why. Glucose is number one. Doesn't matter if you have low lactose, high lactose. Doesn't matter. If glucose is in the presence, so yeah, let me clear this up. If glucose is around, E. coli does not care how much lactose you have. E. coli has that system where it's built in a specific way. Check this out. That if you're having an increase in cyclic AMP, let's say, well, a decrease. Let's say you have a decrease of cyclic AMP because you're eating so much glucose, right? You're eating ATP from the glucose. You're loving that ATP. Guess what will not be stimulated? Guess what cannot be stimulated? That RNA polymerase. Why? Because you're unable to form the complex. Remember that the complex is only going to form when you have a high level of cyclic AMP and the cap complex. No cap? Yeah, basically, if you don't have an increase in cyclic AMP, you cannot bind cap. It must be increased in order to bind cap. If cyclic AMP is decreased, that means that we're eating well. we're eating well. Does that make sense? Good, good, good. So yeah, that's the rule of the game. Ask me any questions. That's how we can best understand this whole idea, this whole process, and we'll do some questions on this right now. The best idea you can have with this is give it some thought. Think to yourself, this is such a nice system because if, for example, I'll give you an example. So let's see if you guys really understand this. I'll ask you guys and then you can answer me. If, for example, I have glucose and I have lactose. So can we say if glucose is high, the lac Aperon will not be activated? There you go. That's exactly what I was about to say right now. That's the example I'm giving you right now, where you have glucose right here and you have lactose. Why in the world is it going to activate the lac Aperon to break down lactose if you already have E. coli's preferred meal? E. coli's, that's like steak. That's like, I don't know, pizza. That's like, I don't know, whatever you guys' favorite meal is. This is so diverse. We're so diverse here. I can imagine we're from all different parts of the world. I'm over here thinking with my American brain. Everybody's favorite meal. Mangú con plátano, salami, basically. That's the glucose. It drops everything and it goes for glucose if glucose is in its presence. So if glucose is there, then forget about lactose because it will start to consume glucose, break it down into ATP, and that high ATP will reflect with a low cyclic AMP. Low cyclic AMP, just like Muskan said, no cap, no camp. Does that make sense, everyone? Yes, Amna, you are correct. It's like a backup plan. No cap. Definitely. 100%. Yes. This is how it's happening. This is how the system works. And yes, just off of this idea, they will try to test you over and over again, trying to see, hey, glucose is there, but so is lactose. Is lac Aperon activating or is this happening? They're just trying to mess with you. But if you understand the system, then you know this. And this is not too crazy. So what I'll do is I'll run it down from the top, step by step, just in case anybody did not understand. And it is recorded too. You guys can look back at the video and watch it over again. But one thing I will say, I would run it down. And if you guys don't understand, write a question and I'll answer it. And if anything as well, we'll do questions right after. Let's see if we really understand. So yeah, we need some participation, some people raising their hand. Got to be brave. Let's do some questions. But first, let's start off. Like I said from the top, lac Aperon is a system. It's a system that E. coli can use in order to consume lactose. Remember, E. coli, its favorite food is always going to be glucose. So glucose, the first thing you need to understand when it comes to lac Aperon is the idea of the energy source. If you are depleted, you will have an increase in cyclic AMP. If you are in a full state, you'll have an increase of ATP. So let's understand. E. coli favors glucose. Okay, so what ends up happening is that in order for you to consume an alternative source of energy like galactose, you have to first break down your lactose. So what's the issue here? Well, the issue is this. You have something called lac-I. Lac-I codes for the genetic material that's able to transcribe repressors. So it makes repressors. And repressors are able to basically block the, let's say, the affinity, let's say, the activation of RNA polymerase. Remember, we also mentioned that prokaryotic cells are polycistronic, meaning that they can actually utilize one mRNA to code for different types of proteins. Unlike us, who are monocistronic, one mRNA, one protein. So they're basically, I'm just telling you that LAC-I is making inhibitors. It's making repressors. Repressors are binding to that O site. And remember, in order to activate your lactose metabolism through the lac-Aperon system, you do need negative stimulation and positive. The positive stimulation lies in the idea that we are in a starvation state, that we are not consuming alternative source of energy, that all this alternative source of energy is not available to us. And how would that reflect? That will reflect with an increase of cyclic AMP. Cyclic AMP binds to something called CAP. That will form a complex, cyclic AMP CAP complex, that wants to stimulate RNA polymerase to code for the structural proteins that are found in the genes Z, Y, and A. We said Z was beta-galactosidase, Y was permease, and we said that the Y kind of looks like an arrow like this. Is grabbing E. coli and injecting basically that lactose. That's a Y, it's a permease. A is an acetylation activity that just detoxifies E. coli. Not too important. Not too important. But okay. Basically, that is the positive stimulus. But this positive stimulus cannot work if we don't remove the repressors. And what in the world is going to remove the repressors? Something called lactase. Or allolactose, I should say. Allolactose is nothing more than a lactose. It's just a lactose that underwent a specific type of reaction from the enzyme beta-galactosidase. And this is where it got a little confusing. Because it's like, hey, what in the world? We don't have beta-galactosidase because we're not able to break down lactose. And I mentioned to you, I said, hey, we do have a minimal amount, something called leaky activation, minimal amount. And this minimal amount favors the activity of conversion of lactose to allolactose. That's what it's favoring. It's not even favoring the breakdown. But once you're able to then remove that repressor, then you will have lac-Aperon stimulation. Now you will have mRNA binding to the promoter site, mRNA coding for ZYA. Now you will have Z. Z is galactosidase. Galactosidase now will increase in number. And when that increases in numbers, now it's breaking down lactose, hydrolyzing it, turning it to galactose and glucose, consuming it as fast as it can. Hopefully that makes sense, right? Any questions? If not, we jump into actual question, okay? All right. So show of hands. Who wants to go? Who wants to go? Who wants to do this question? Who is the brave soul here who wants to do this question? Who is brave enough? Oh, yeah, definitely. Muskan, definitely. Please give me questions. I want questions because I feel like if you guys ask me questions, there's probably somebody else who's not asking. And then that person, you could also clarify any kind of doubt that they have. Yes, Muskan. An increased breakdown of lactose to glucose and galactose. Yes, that is correct. That is correct. Which will be used by E. coli for metabolism. Yes, that is correct. Definitely. That sounds perfect. Yes, that's what it is. It's simple. I'm telling everyone right now, this is a very simple type of activity. The only thing is that they're very creative with the questions. They're very creative with the questions because they're going to dive in. So a good strategy for approaching any kind of lac-Aperon question is to first identify in your mind, am I in the presence of glucose and galactose? Let's not fall for their tricks. Let's reverse it on them. These are their favorite questions. They like to ask a question based on a patient who is in the presence of both. So both glucose and lactose need to be low for E. coli to work. Low glucose, low lactose leads to allolactose, remove repressor. Okay, so yeah, let me clarify that really quick. So it's not the low lactose that leads to allosteric activator. It's not the low lactose. What I'm saying is that the low amount of that enzyme, beta galactosidase. It's not really the amount of lactose. It's not the amount of lactose. It's just that the enzyme is so low. They call this in physiology, this is a weird phenomenon, but they call it a trickling phenomenon, where there is minor transcription of this specific enzyme. And supposedly the theory behind these kind of trickling activation, trickling transcription, the idea behind it is that if, for example, you get overflow of, let's say, any kind of energy source has an overflow, like let's say you have a whole bunch of lactose, you have to have somewhat of that enzyme there in order to support the patient, because you can't just have a whole bunch of transcription. You can't be dependent on that transcription right there, like just that on its own. That's the theory behind it. I read up on it. It's kind of weird. Look it up on your free time. It's trickling transcription, basically. So let's see. She also says, I have a question. Beta galactosidase only breaks down lactose to allolactose when there's high lactose level and low glucose. Yes, it breaks it down. I'm not going to tell you high lactose levels. I'm just going to say that when it's in the presence of lactose, yes, it turns it into allolactose when it's in the presence of lactose. But this is the thing. You can turn lactose into allolactose all day. You can do this all the time with glucose being there. But it will not stimulate the mRNA. It will not stimulate the RNA polymerase. Do you know why? Do you understand why? Let me see if you understand this. What I'm telling you is you can have beta galactosidase stimulating lactose, turning it into allolactose. You can have this happen, and allolactose will remove the repressor. It will remove the repressor that's found on the Operon. But yet you will still not have stimulation of the RNA polymerase. Do you know why? It will not do it. Yes, because you are in the presence of decreased cyclic AMP. Does that make sense? In the presence of decreased cyclic AMP, you cannot bind CAP. No CAP complex. Yes, there is no CAP complex formation because your CAMP, your cyclic AMP, is low. Yes, perfect. So it doesn't matter if they're turning it into allolactose. Doesn't matter. Because it doesn't have the positive stimulus. You have to have both remove the negative stimulus and stimulate the positive. Does that make sense, everyone? Two things. Yes. Perfect. Let's see. Yeah, we got some good questions here. Let's see. Low glucose or no glucose means low camp binds cap. So I'll correct that. I'll correct that for you, Sandy. Low glucose or no glucose means high camp. And that high camp binds cap. So here I drew this showing an example of how this will not be able to bind because it's low. When I drew that, that was an example of how it cannot bind because it's low. So normally when you have cap, you need cap and increase cyclic AMP in order to form this complex to start off the lac Aperon system. Let's see, low galactose side produce allolactose and eliminate promoter. Not eliminate promoter, they eliminate repressors. So yeah, let me be very clear with that. So two things have to be in sync, Carrie says, like you said in the start. Yeah, you need to have both positive and negative stimulation on lac Aperon in order for it to transcribe lac Z, Y, and A, perfect amna. That's it. That's exactly what it's about. Keep that in your mind. Alexandra says, so only when camp is high, it binds to cap. Yes, 100%, Alexandra. That's what you have to keep in mind. That cap complex only happens when cyclic AMP is increased. So yes. So Sandy, let me put it out. Let me write it out for you like this. When we have the lac I, remember that the lac I is the one that codes for repressors. Repressor is going to bind to the O site, the Operon site. Remember that the promoter region is here, the P. In order to remove this repressor, you need to have allolactose. So that means that lactose needs to be within the presence. I knew that the little beta galactosidase thing was going to confuse people. Yes, because it is confusing. It's like, why in the world is it this enzyme the one doing it? I wish it wasn't this one and it was another one, but it's this one. But it's only here because of a trickling effect, trickling phenomenon. Read up on it. It's kind of crazy. But yeah, allolactose will bind this repressor and remove it. That's how it's working. That's its mechanism. When it removes it, now you have your RNA polymerase able to transcribe ZYA, able to make your mRNA code for these specific enzymes. The permease activity from the Y, beta galactosidase now increases. And when beta galactosidase increases, guess what happens? No more prioritizing the allolactose because allolactose is already binding it. It's already at full capacity, has binded all the repressors. Now we are caring about breaking down the lactose. And this only happens when glucose is low. Yes, you are right. Only when glucose is low. It's like E. coli is getting picky. It's like one of those babies that you take out to, I don't know, eat and they don't want to eat some foods because they want McDonald's or something. They just don't want to eat. That's how it is. It likes its glucose. But when it realizes that there is no other choice, then it must consume lactose. Right? It must break down the lactose and consume it. And it'll only consume it with two steps. Remember, you have the removal of the negative influence and then the stimulus of the positive complex that would force it to transcribe. So yeah, that's basically it. It's not tough, guys. It's not tough. Not tough. So this is a good video you guys can always come back to. Just look at the end of anything and just listen to me run it down from beginning to start. I basically run it all down. So, okay. I think we're ready for questions. Who is brave and wants to do a question? Oh yeah, any questions? If you guys have any more questions, let me know. If you guys have any more questions, let me know. Let me know. definitely will answer. Let me know. Okay, question time. Here we go. Here we go, guys. Let me start a new screen so that way I don't have any lagging on my screen. So yeah, this is what I basically do. I have a Patreon class. We give class all the time. We're just basically giving class on a whole bunch of different subjects. This month was pharmacology month. So yeah, we just give class all the time. And yeah, this is today's class basically. I told my group to join today as well. So I'm interested to see who gets this next question right. Well, who understands it? It's the better way for me to say it, actually. Who understands this question? So here he goes. Okay, we got Dr. Carey. Dr. Carey, let's go. Let's go.
Okay, let's see. Which of the following is the most likely explanation for the observed growth pattern? Okay, the graph shows the growth of E. coli in culture media containing glucose and lactose. Okay, so basically, like you said, if glucose is present, right?
Yeah.
Glucose is present. So let's see what the lack of operon is not going to be activated.
Correct.
So altered non-functional catabolite activator protein, glucose depletion followed by induction of the lac operon, that is not going to be. Increased beta-galactosidase synthesis in the first 4 hours of growth. Okay, so I see relatively growth in time, but I don't know here what is growing. So lactose depletion followed by glucose utilization. Increased beta-galactosidase synthesis in the first 4 hours of growth. I mean, that could happen because it's increasing.
All right. One second before anything, Carrie, I want to tell you, can't fall for their tricks. They in certain type of questions, in certain types of like biochem is one of them. I'm not going to say all, but there's certain things where the option choices are used against you. It's like weapons that they just turn against you. So it's not like this with all subjects, but with this one, what you would always have to do. So I'm going to give you this quick advice. When it comes to molecular biology, when it comes to biochem, when you read the question, the first thing you should do, analyze and think, what pathway am I on? What is the goal of this pathway? In this case, it's a bit different. In this case, it's like, okay, how does E. coli respond to the consumption of glucose and lactose, and how is this timeline relevant to that? Try to make sense of that first.
Okay, so what I'm thinking is that E. coli is just going to start depleting glucose. Okay, because that's what they're going to favor from the beginning.
Yes, I agree with you. I agree with you there. It's the number one desired food, right?
So I think it's going to be glucose depletion. Once they deplete the glucose, they're going to follow by the induction of the lac operon, because then they will deplete the glucose.
That makes sense, right?
Yep.
So what answer choice represents that?
B.
Yes. Good. Muscan. Yes. Definitely. That's what's going on here. So let's look at this. So notice that the y-axis here says relative growth. Relative growth. So I did mention in the very, very beginning of class, I mentioned one thing. I mentioned that bacteria likes to thrive, and it thrives when you either eliminate its competition or when you feed it. So here, let's see, lactose depletion followed by glucose utilization. Yeah, you got to remember, glucose is number one. Glucose is like
that would be the opposite.
Yeah, definitely. You got to remember, always, glucose is number one. So check this out. Here, you'll see rapid growth when there's more consumption of food, when you're giving it more food, when it's able to actually break down its food. So here, when I see this, I'm seeing, okay, let's see what's going on. So yeah, Carrie, you hit it the nail on the head when you said that at first, the manifestation will represent itself as the consumption of glucose, right? You would first consume glucose, let's say the first 4 hours. That's the number one source. You know what? Look how that jump, how big it was. This is because it was consuming glucose. Now, as the glucose started to deplete, as the glucose started to get low, what started to increase, everybody? You tell me. Type it in. What started to increase as glucose started to get low? What started to decrease? To increase? Increasing cyclic AMP. Yes, 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 yes. Type it in so that way you don't forget. Cyclic AMP increased. Increased. Okay. Now I'm going to ask you guys another trick question. What else started to increase? What else started to increase? Activity of adenylate cyclase. Okay. Yeah, you can say that. You can say that. Cap activity, yes. Cap complex started to basically react. Yes, you have cap complex. Cap complex. And okay, that's good. That's good. Yeah. Let's put it like this. These two things they did start to join and form a complex. Yes, Maria, allolactose. Allolactose now is reacting, and now it's removing. You're going to have that removal of the repressor. Now, where does the repressor sit? Where is the repressor sitting? What's his favorite chair? Where does it hang out? Where is its place? Where does it stay? On the O, right? On the Operon site, yes. That's where it likes to stay, on the O. So you have to remove it with allolactase. And once you remove it, now look what's happening at the 6th hour, at the 8th hour. Look at what's going on here. Here you're having induction of lac-Operon system. This is not bad. I'm telling you guys, this is not bad. This is understandable. We got this. We know this, right? Not crazy. Yeah, let me see. Let me see if let's see what we got. See what we got here. Let's see. We have something else. Nah, I doubt. You know what? Let's see. Let's see. Let's see what's up. I think we could have another question here, actually. Let's see. Let's see. Let's do another question. Who's next? Who is? That was a good job, Carrie. Good job. We have Amna. First come, first serve. We have Amna. Let's go, Amna.
Okay.
Let's go. Let's go. This is straight up Amboss.
Okay. So the observed results are most likely due to which of the following properties of broth A compared to broth C? Okay. So an investigator inoculates 3 different broths with one colony-forming unit of Escherichia coli. Broth A contains 100 micromole of lactose. Broth B contains 100 micromole of glucose, and the broth C contains both 100 micromoles of lactose and 100 micromoles of glucose. Okay. After 24 hours, the amounts of lactose, galactose, and glucose in the 3 broths are measured. The results of the experiment are shown. Okay. So in broth A, we had lactose. Okay. So now they are measuring lactose, galactose, and glucose in all of these 3 broths. Okay, makes sense. So yeah, in broth B, we only have glucose. Since glucose won't be broken down into lactose and galactose, makes sense that broth B has 0 lactose and galactose. Okay. So are likely due to which of the following properties of broth A compared to broth B?
The observed results of the following. Let's see. This is okay. The observed results are most likely due to which of the following properties of broth A compared to broth C. So they want you to compare A to C.
Yes.
Okay.
Okay, so in broth A, we had lactose, whereas in broth C, we have both lactose and glucose. Okay, so when observed in the results, broth A with the lactose, it's 43, and broth C, lactose is on the higher side, whereas the glucose is 66 and galactose is 1. Okay, so they want to know that what has happened between broth A and broth B. Okay, so the lactose in the broth A is 43, whereas glucose is 9. Okay, whereas broth C, glucose is 66 and lactose is on the higher side. Okay, so this means, yeah, it does make sense that in broth C, the E. coli have used glucose. That's why it has dropped to 66, where I'm talking about the broth C.
Okay. Broth C.
Yeah, and whereas the lactose in the broth C is 94. Well, in broth A, it was only lactose that was provided to the glucose. So yeah, it does make sense that the E. coli has to use lactose because glucose wasn't there in broth A, right?
Yeah, in broth A, right? In A. That makes
Yes,
sense.
yes. That's why it has tremendously dropped from 100 to 43. Okay, so I'm gonna look at the options. Increased activity of glycosylases. Okay.
Do you remember what glycosylases? Well, no, this is not even. Yeah, let's not even jump into that. That was something completely different.
Yeah,
That
I
has to do
remember
with the
the
DNA base
lysation.
repairs. Yeah, that has to do with the base repairs. Remember, they were breaking off a chunk of the DNA in order to repair it. That's something completely different.
Okay.
Remember the DNA repair?
Yes, I just remember
Yeah,
now.
yeah.
Okay, so decreased activity of catabolite activator protein. Okay, the activator protein, the one we had, the it was CAMP, right? Yeah. So
Also known
decreased
as CAP.
activity. Yeah. Okay, decreased activity. So first of all, they want me to tell that what's the difference between broth A and broth C. So what I know is that definitely E. coli in broth C, E. coli will first act on glucose and then on lactose. But since they are utilizing glucose, that's why glucose is dropping and lactose is 94. Whereas in broth A, it's only the lactose that is present.
So
Okay.
basically, this is what they want you to know. They just want you to tell them that the difference that you're seeing here is due to what property? What is the difference? So this is how I would do this before anything. Yes, definitely, definitely. Yeah, Muskan, everybody, let me tell you guys, let's give it a little pause here. Let me just tell you guys a good strategy. We have to strategize always. Don't ever do what we're doing today. I'll tell you why. When you're approached with a molecular biology question or a biochem question, I'm always going to try to steer all the students away from this technique. The technique of trying to find the answer in the answer choices is dangerous because the test writer can write a piece of a correct answer and then end it in an incorrect way and confuse the crap out of you. Now you'll be sitting down thinking, whoa, this is right. But at the same time, it's kind of wrong. But it looks better than this one and it looks better than that one. Now you're fitting your answer to a puzzle piece. What you should do first, always with molecular biology and biochem, this is not with all the other subjects. On this one in particular, you should think to yourself, Okay, what are they asking me? They're asking me about lac-Aperon. They're asking me about A and C. Look how easy this strategy makes it. Watch. All I have to do is this. What's the difference between A and C? A is lactose. C is lactose and glucose. Okay. Using my knowledge now, before even looking at the answer choices, using my knowledge, what happens different in C in comparison to A? There's something different that happens here.
Yeah.
So you tell me.
Yeah, so the bacteria will utilize glucose, and once the glucose has depleted, then the bacteria will move on to lactose.
Okay, good. And when it uses glucose, what is high? What increases?
So it's the CAMP that gets high.
It gets high once it's depleted, right?
Yeah.
So remember, at first, so let's put it like a spectrum. I'm going to write it right here like a little spectrum here so you guys can look at it like this. Let's say we're talking about the utility of glucose. Glucose utility. In the very beginning, your cyclic AMP is going to be low. Let's say you're having high level of glucose. At the very beginning, you're having a very high level, and then as it starts to deplete, as you get lower, your cyclic AMP increases, right?
Yeah.
Like a spectrum. It's like a spectrum. So here I'm going to put it high versus low. I know it looks crazy, but I'm going to put this in the chat just to remind everyone to keep this in mind. So we'll look back at the questions that everybody's writing. But for now, yeah, let's go, Amna. Definitely. So what I'm asking you was this. What's the difference? You said that they have to consume glucose first. That is true. They have to consume glucose first. And when they consume glucose, you have a decrease of cyclic AMP in the very beginning. Then towards the end of the usage of it, it increases. After it increases, what happens? You form the complex,
So we
right?
form this complex, yeah, with CAP, and the other thing that has to come is the removal of the repressor protein. Yeah.
Yeah, and it does happen different. Well, it happens the same in both of them, right?
Yes.
Because they both have lactose. So they would both
Yes.
have the negative activity. And you remember the negative activity? What happens?
Yes. So we have the allolactose that basically comes from the lactose in the presence of low amount of beta galactosidase. Yeah, and we talk about the trickling phenomenon where there is this transcription of beta galactosidase happening. And this is how the allolactose will bind to the repressor protein and will remove the repressor protein from the oocyte.
Perfect. Yeah. Perfect. That is completely perfect. That is exactly what we said. That's like photographic memory. That's perfect. That makes 100% sense. Remember, you're removing the repressors. You're removing the repressors on the oocyte. Yeah, perfect. Perfect. So we understand that. We have a good general understanding of everything. That's great. Now, this is how I approach the question. I understand that. That to me is in my head. I know this. That's the difference between both of them, because we said that they are different. You have A and then you have C. So
Yeah.
I can tell you that A will not have.
Yeah, the only thing that will be differentiating them will be the increased CAMP.
It from, yeah, that will be differentiated, right? Yeah. So let's take a look at this.
Okay. So decreased production of, okay, so it's not B. If we talk about C, it's decreased production of alpha galactosidase A. Okay. We
That's
did talk about,
that's a crazy answer. That's lysosomal storage enzyme. That's They're going the lysosomal route on that one. That one is crazy. That's Fabry's disease, I think.
Yeah.
Yeah.
The one we have talked about is beta galactosidase, right?
We talked about beta galactosidase. Yeah, they're trying to mess with you here. Alpha galactosidase, that's when you have Fabry's disease and lysosomal storage. That's
Yeah.
completely different. Yeah.
Okay. And then we have the increased activity of adenylyte cyclase. Okay. Yeah, it's the enzyme that basically changes ATP to CNP. Okay. So I'm gonna keep this for now. Binding of repressor protein to the operator protein. Yeah. So over here they're talking about the allolactose. Yeah. So this is the step that's common in both of them. Yeah, I'm gonna go for D.
Okay. D, you say, right?
D.
Okay, good. That is the answer. That is the answer 100%. Yes. Yes, yes, this is the answer. And this is what I'll tell you. Yes, the answer is D, everyone. The answer is D. I will look back at your questions. Anything that anybody asked, we're going to answer them. Definitely. The answer here is D. Now, why does D make sense? Because glucose will cause this type of reaction. You're going to have an increase, basically an increase in CAP, right? You're going to have that increase in the CAP system. You will have an increase in that CAP and increase in cyclic AMP. That's going to happen in that sense. So this makes sense. This makes sense because that's the difference between both of them. That's the only differentiating detail here is that glucose is putting in that work that I just sent you guys right here. The whole idea of, it looks like a crazy racing car, but this is what we mean right here. But yeah, the decrease in cyclic AMP, you're seeing that going to an increase in cyclic AMP when you utilize your glucose, when your glucose is low. And that's what activates the CAP. That's what triggers increase stimulation. The promoter sites basically have enhancement, and then your RNA polymerase is able to bind the promoter, and then you're able to transcribe Z, Y, and A. Now let's look at the questions that you guys asked. I'm very interested in your questions. Let's see. And okay, so somebody said something. Okay. And this is only happening when glucose is really low. Cool stuff. Okay, cool. Let's see. Doc, which part of E. coli does CAP act on? Missed that part. Okay, let's talk about that. So E. coli is a prokaryotic type of cell. Unlike us, we have helical type of DNA. They don't. Prokaryotic cells have circular type of DNA. It's a circle. So when they replicate, they have one origin site. So they only open up in one area. And then they have their DNA-dependent RNA polymerase binding, able to basically replicate. But that's not what we're talking about. This is not replication. When you look at prokaryotic cells and their ability to transcribe mRNA, instead, they can just enhance several parts of their genetic structure in order for this genetic structure to then expose itself. And you'll have transcription factors, you'll have enhancers being able to allow promoter regions to stick out. If promoter regions are sticking out, this is in the nucleus, by the way. If the promoter regions are sticking out, then you're able to have RNA polymerase bind to them. And when they bind to it, they're able to transcribe a complementary strand of RNA. Remember, RNA uses uracil. So you're going to make RNA, and it's going to come out as an mRNA. mRNA is able to then leave the nucleus. Remember that mRNA does not do any of that crazy stuff with capping, poly-A tail, and spliceosome. We do that as eukaryotic cells, right? So prokaryotic cells, they give them a break here. You're able to get it out of the nucleus and put it into the cytoplasm. Once you put this into the cytoplasm, you're going to find ribosomes. Yeah, don't get me started with ribosomes and molecular biology. We did a camp not too long ago, so I have everything fresh in my mind. mRNA, remember guys, mRNA has a shindogonal sequence. A shindogonal sequence. And it's going to bind the 16S of the 30S. And it forms this complex right here. 30S, 60S. This is how translation happens. So first we transcribe in order to get mRNA. We transcribe, transcription. That's what it is. This mRNA, yes. So to take a listen, guys, this is how it goes. mRNA transcription. mRNA is going to come from a product of transcription. You were able to transcribe this because you had expression of that gene. That gene was like, hey, I'm here. You're trying to make a protein here, you could bind. So RNA polymerase was able to bind that promoter site and then code, basically make the complementary of that strand. Once it made the complementary of the strand, it made an mRNA. That's called transcription. The next step is translation. Translation is when you take that mRNA protein that you just made, that sequence of nucleotides, let's say C, G, oh, there's no T's, there's U's instead. G, C, U, all this sequence, whatever in the world the sequence is. You take this sequence and you're now trying to make this sequence into actual proteins. In this specific case with E. coli, the proteins that you're going to make, we mentioned them already. Galactosidase, permease, and acetylation. That's the type of proteins that you're going to be making here with the ribosomes. And remember that the first step, remember you have initiation, elongation. Yeah, let's go through that. I don't mind at all. Let's go. So pay attention. I'll tell you right now, pay attention. Put that mind into focus. Let's go, guys. Let's go. So yeah, I'm always going through this with my Patreon group. My Patreon group must be tired of this. You guys must know everything that has to do with transcription because this is what we do all the time. So if anything, yeah, look, everyone is like, oh my God, come on, because they know this so well at this point. I'm repeating this all of the time because this subject always falls into my hand. It falls into my hand in micro, falls into my hand in biology and in molecular biology. It falls into my hand in pharmacology because so many drugs can work here. So let's take a look at this. All right, mRNA has just gotten, yeah, you got me started. That's it. mRNA has just gotten transcribed. mRNA makes its way outside of the cell. mRNA has here a specific type of sequence called the shindogonal sequence. Shindogonal sequence is actually complementary to an rRNA that can be found on the 30S. This 30S has a small little protein called 16S. So shindogonal sequence is actually able to bind this 16S. This is the very first step of translation. This is called initiation. This is just starting. Yes. It's like, hey, come over here. We got you right here. Don't worry. Come here, bind here. And it comes in and it binds. It actually binds. Okay. So let's show you. That's the very first step, right? You get those together. Now, the whole mission here that we're trying to do when it comes to translation is three things. We want to do initiation, we want to do elongation, and then we want to terminate. Okay. So let's understand this properly. So number one, yes, you are bringing your mRNA onto 30S. Look at it right here. We're doing this. mRNA shindogonal sequence going to the 30S. That's step one. Let me tell you guys, there's a drug that inhibits this very first step. My class knows. I don't even have to ask them. Aminoglycoside. Aminoglycoside. How do I remember this? A is the first letter of the alphabet. Aminoglycoside is the first letter of the alphabet. Is A. That's the very first step in the initiation. You're blocking the initiation complex. You're binding the 16S portion of the 30S in order to block mRNA. That's the very first step. Next step. So what happens now? Now you're having your mRNA on top of that 30S. Now here comes the problem. We have regulatory types of proteins that are located here on the actual ribosome. They're regulatory types of proteins. They can regulate the next step. You don't want to advance. You want to stay step by step when it comes to translation. So let's be very clear. For example, as the mRNA displays itself here on this 30S, you're going to realize that it's going to be able to express a start codon, AUG. This codes for methionine. But notice how it's not expressing any other types of codons. Let's just give it a GGG. It's not expressing these type of codons. Why? Because you have something called initiation factors. They're looking out for you. They're making sure that you don't skip any steps, that you go step by step correctly. You don't mess this up. So initiation factors, every single time we talk about the next step, whenever you hear me say the next step, I'm going to tell you that initiation factors basically have used GTP in order to get out of the way. To get out of the way. They need to get out of the way as we advance. That's the name of the game. Anything that ends in factor is using GTP. So mRNA has binded the 30S via that 16S part. Guess what ended up getting hydrolysis reaction with GTP? Can you guess? An initiation factor. It's not important to know the initiation factors. I'll tell you which is the only factor that is important. It's an elongation factor, and I'll mention it. But so far, yes, this has entered. Now, what's the next step? As this is happening, you need tRNA to help you out. You need tRNA in order to be able to actually basically build up your polypeptide protein. So tRNA has to come in and help you. It has to grab an amino acid, and it has to also enter this site. So AUG codes for methionine. This is always going to be the same start codon. This is always going to be the same amino acid. So just letting you know, the tRNA has an arm, has two arms. It has one arm here. It has the T arm and it has the D arm. Yes. Wow, you guys know it so well. The D arm is recognizing amino acyl tRNA synthesis. Synthase. This is going to recognize an enzyme that is responsible for phosphorylating the amino acid that tRNA will pick up. Now, how does it know what amino acid to pick up? Because it has anticodon right here. It has anticodon right here. The complementary of whatever is being expressed on that 30S. T-arm is for tether. So this tethers it. This brings it over to the mRNA. I'm sorry, to the, the mRNA, the complex that we see here. And now let me also tell you, this phosphorylation process happens utilizing ATP. ATP will actually catalyze this reaction where now your amino acid has gotten phosphorylated. That's called charging. So if they say, hey, is this amino acid charged? Yes, it is. It just got charged. So the amino acid will make its way here and it will bind here on the P site. So you have to write here ape backwards. I say arriving, printing, and exiting. Only the start codon arrives on the P. That's it. All the others will arrive on the A site. Okay, so let me continue. Let me just finish this off real quick. I'm just going to tell you what ends up happening is that once this enters here, we can't forget about our other piece, the other piece, which is very important as well, the 50S. The 50S needs to come in as well and join the complex. Then finally, you have the initiation complex formation is full, is complete. The 50S comes in. It also joins. So at the end of it all, it should look like this. This looks like a little messy here, but it should look something like this. Looks a little messy, but here you have your 50S. You have your tRNA. You have your 30S. And you have your mRNA. That is all the components of the initiation complex. Once this is all together, now you're done with initiation. Now comes elongation. Elongation. You will now express the next codon on the A site. A is for arriving. So what ends up happening? Now a tRNA will arrive at the same site, at this site, with the anticodon in the bottom, with the actual amino acid nice and charged, bringing it to the A site, bringing it into the A site. All right, so what happens? When you bring it to the A site, you're going to now have, look at how weird it gets. I'm going to draw it right here. It gets super weird because now you have two. Think about this. Now you're having a T here, tRNA with an amino acid, and a tRNA here with an amino acid. They're different amino acids though. So now you have to transfer the amino acid on the P site. You have to now transfer it to the A site. The new tRNA will always get the old amino acid. This happens with an enzyme called peptidyl transferase. Peptidyl transferase. We could inhibit this enzyme with chloramphenicol. Phenicol. Okay, so you're transferring that. Now, once you transfer it, you have activation of something called elongation factor 2. Elongation factor 2 will stimulate the next process called translocation. Everything will move over one side. Everything moves over one side. So now your tRNAs, instead of them being here on the AP site, you'll see that your tRNAs are now moved over into your E for exiting and your P site. And the P site has the two of them. It has the two amino acids. Now the A site is exposed, and then another tRNA comes in, and the story continues until you start building more and more amino acids, until you end up reaching a code that would code for a stop codon. You are away, you go away, right? You remember those mnemonics? UGA, UAA. Yeah, all that would code for stop codon. That's it. We're over. We made our amino acids. We're ready now. So then they have releasing factor, which will release them. Once you release it, then you have your protein. Your protein is made up of a chain of amino acids that you have made yourself. So the wobble hypothesis applies. If, for example, the tRNA is bringing, let's say, anticodon material in the bottom, right? It's bringing an amino acid that it code for. So I would have to show you the wheel. There's a wheel that you'll see. And this wheel shows you, let me show you this wheel actually, because you could actually mess up. Remember that when we talk about anticodons and codons, we're talking about pairs of 3 nucleotides that should code for a specific amino acid. So they are allowing you to mess up on the last nucleotide and still bring you the same amino acid. That's the wobble hypothesis. You're able to basically have some kind of forgiveness. That's how I see it, as a forgiveness, where if you are coding for the wrong nucleotide on the 3rd site, you're able to still code for the same amino acid. That way there won't be a mutation or a problem in that sense. That's how tRNA is trying to help you. So let me put the nucleotide wheel. Let me see. Let me show you guys like this. It's pretty cool. If you understand it like this, show you guys a little image. It's pretty cool. I love it because if you look at it like this, Look at that. For example, if I have a code that says G A G, that's coding for glutamine, I think, or is it glutamate? Glutamine. Glutamine. Notice how you could actually have a different code in the end and still make your same kind of amino acid in that sense. So when we look at, let's look at an image, wobble phenomenon. Yeah, I don't want to confuse anybody. I'm confusing people. Let me see. The tRNA wobble. Yeah, let me show you guys. Yeah, some tRNA molecules are able to recognize multiple codons and they're able to wobble. So they basically, yeah, let's show you an image. I think the image will be nice right here. Yeah, it does it with 3 different types of nucleotides, which is pretty cool. That way you don't make a mistake. That way it protects you from making a mistake. So this is straight up from Amboss. I think they do a great job because of the image. Notice how this last one here can fit into any of these type of nucleotides, and it will still code for the same type of amino acid. It's recognized. Codes for the same amino acid. Does that make sense? The third position, basically. They call that the wobble position. So it uses an inosine for it to be able to nullify whatever damage or whatever problem would have happened here. So yeah, it has, that's basically, if you think about it, it's just an idea of flexibility of, let's say, the lack of leniency. I would call it lack of leniency in base pairing. Base pairing forgiveness. That's a nice way to put it. Forgiveness. So yes, that makes sense. That makes sense. So yeah, guys, this is it. This is what I do. I'm giving class all the time. We just gave 2 hours right now. This was good. I hope everybody understands the lac Operon. Definitely. Anybody, if you want to take a lot of classes, you can always join my Patreon. Patreon is active. I'll definitely put the link. We're giving like 16 classes a month all the time. Definitely. Also want to thank Dr. Lopez giving me the opportunity definitely to give all this class. Yeah, definitely. I also take topics on request. I also tutor one-on-one. Let me put here my Patreon page. It's just patreon.com/Dr. Batista. That's what they call me, Dr. Batista. So if you guys have never seen me before, let me know because I'm always giving class. I'm always around. Definitely. I don't think I've seen you before, Muskan. Never seen you before. But I love the participation. That was a good class. I love the participation. I love how everybody participated. And I think everybody understood. I think everybody, for the most part, understood. So that was pretty good. Okay, perfect. Yes, the oncology one. Yes, I remember that one. That one was kind of crazy. We went into lac Operon. Definitely. I remember that. So, let's see. Okay, yes, definitely Haiti. Nice to meet you. Nice to see you. What I usually do is drills. I don't really give lectures to Lopez class. Usually I'm here giving drills. Basically what I do is I jump into a subject and we just start going crazy. Like today, I can just freestyle a lot of subjects. So if you ask me a subject, I can just go into it. And we can just start talking about drugs and micro and neuro and all that. So yeah, I do a lot of drills. I'll probably do more drills this month as well. I'll be sure to tell Lopez if anything, and we can do drills as well, guys. Definitely. Pharmacology is what I was doing this month. So in this month, I'm going to finish up strong with a lot of class. So yeah, I'll probably get another drill in. I'll talk to Lopez. Definitely schedule 1 drill at least for the month. Definitely. Yes, Jay. Yes, yes. Thank you for attending. Thank you, everybody, for attending. Definitely, definitely. This will be recorded. You can go back and watch this again. Um, yeah, I did enjoy it. I did enjoy it. I think the question really put everything into perspective, though. The more, the more I see it, it's like the question really like let us know, hey, do we know this subject or not? Because they're going to test us. They're going to test you to see if you know it. It's cool to know a whole bunch of stuff, but you got to put that answer correctly in your test. That's what counts. That's what counts. It's the answer, the right answer. Yes. Yes. Definitely. Definitely. So thank you, everyone. Thank you, everyone. That's my Patreon page. I think I put it out there. If you see it, click it, join it. I'm also on Instagram. I'm basically everywhere. I just gotta promote myself a lot more. Not promoting myself at all, but yeah, I'm always just giving class busy. So definitely thank you everyone for joining. Bye. Goodbye. I'll see everybody either on Patreon or yeah, you can also join my Instagram. I'm also posting mnemonics and everything there on Instagram. Definitely, definitely. WhatsApp as well. We have a little WhatsApp group going on. We also give class. Definitely, definitely. I have the link to the group in the Patreon page. It's like the first thing that you'll see there. So yeah, thank you everybody. Everybody have a nice day. And today's anybody from the Patreon class, my Patreon class, this was the class for today. I put it at 4 o'clock. Yes, definitely, definitely. Today's class was this class. So this is my Patreon class for today. So yes, definitely. I'll be posting this as well. So yes, this was a great opportunity. Mariam. That's not Miriam. That's Mariam. Okay. Yeah. What's up? What's up? What's up? Everybody saying what's up in the chat. What's going on? Okay. All right. So yeah, I'm gonna let you guys go. I'm gonna upload this video. It was fun. I'm gonna go eat. That's what I'm going to do. And drink a whole bunch of water. Just finished. Just talked for like 2 hours straight. See you everyone. Take care. Goodbye. Thank you for joining. Thank you for joining. Thank you everyone for joining.